thanks everyone for taking the time to join us. So for the first 15 minutes, I just wanted to give an overview of kind of our lab group, kind of what, give you some examples of the kind of studies and measurements and things that we do in the lab to get at high elevation pine physiological responses to stress. In particular, I want to highlight each of our grad students' work. So I'll take some time towards the end and highlight what everyone's doing, because we've got a lot going on, as you'll see, and then segue into Lou's specific work. Okay. Start. That's okay. I'm just I'll just put it down here. Yeah. To start, before I forget, I have lots of thank yous and acknowledgements to all of the folks and funding agencies that made all of this work possible. Lots of different funding agencies, fellowship agencies, through student fellowships. A lot of help. It's definitely takes a village to do all of this work. And so thanks to grad students, undergrad students, all of these agencies, and collaborators, and I'll kind of highlight those as we go through. Okay, so as we know, plants, trees, forests, critical and crucial for health, clean air and water, food to eat, awesome outdoor spaces to spend time in. And so given that and the value of forests to all of those things, the goal of my lab is to understand, mitigate, and predict the impacts of environmental stress, both abiotic and biotic, on plant function or plant physiology, and how that dovetails into survival and mortality. And so we do that by quantifying physiological stress or response to stress on the leaf level, whole plant, and landscape scales. And so this, that kind of overarching theme is kind of seen throughout kind of three main research thrusts in my lab: the seedling ecophysiology of high elevation pines, mature tree ecophysiology and kind of forest hydrology that we'll talk about today, and then also plant microbe interactions. But today we're just going to focus on these high elevation pines. All of this work would not be possible without these awesome grad students, and whose work I will each highlight as we go through. Okay, so our main species that we focus on are whitebark pine, limber pine, and Great Basin bristlecone pine. We'll hear a lot about whitebark pine and limber pine from Lou, so I will not go over those. But generally, they're all 5-needle pine species. They're white pine species. They're slow growing. They're long lived, and they're also declining, as a lot of forests are in the Western U.S. Decline due to a suite of factors, including pests like beetle and white pine blister rust, but also wildfire. And so to combat the decline, a restoration strategy, at least for whitebark pine, that was recently listed as threatened under the Endangered Species Act, is to outplant rust-resistant seedlings. And so the seedling is the most vulnerable developmental stage. They're small, they're most vulnerable to climate stressors like drought, heat stress. And so it's critical that we understand how these seedlings will respond to changing climates to make these restoration strategies successful. And so that's the main motivation for focusing on this juvenile seedling stage. And so for a lot of our work, we've obtained seedlings or juveniles, 2-year-olds, 5-year-olds from the Forest Service, from the Coeur d'Alene nursery that grows these species from seed in the greenhouse, and then ship them here to us. And so this is a former master's student, Chloe Wassenes, in the Plant Growth Center greenhouse where our seedlings are today. So we focus on that kind of juvenile stage. And so I'm going to tell you about kind of two studies and overarching takeaways, and then highlight some student work. So the first study is we wanted to first compare the physiological traits of whitebark, limber, and bristlecone. And so this is kind of what they look like. We measured a suite of physiological traits, morphology, stomatal traits, phenology, tolerances to heat and drought, highlight to cold. And one thing that we're working on currently in a paper that I need to get out the door is comparing these traits among these 3 species. And so we have whitebark in blue, limber pine in yellow, and then bristlecone in pink. And so we kind of dumped all of the traits that we have and kind of wanted to pull out what were some top traits that kind of distinguish these species. So we were able to identify some 4 traits that kind of distinguish these three species. And kind of a newer, PCA is kind of a newer analysis to for physiologists. And so one advantage of the study is that we had so many traits, and we wanted to see, okay, could we maybe condense all of those traits down to one dimension using PCA? So that work is ongoing, comparing those juvenile traits among those 3 species. The second study I want to give you an overarching view of is kind of one that helps set up Lou's work that she'll talk about today, is the direct comparison of whitebark pine and limber pine juveniles in the greenhouse. And what we wanted to do is connect these traits, not just compare them between species, but relate them to their contrasting elevational distributions. So Andy kind of hinted at this, that both whitebark and limber pine co-occur in the GYE, but what Andy's work has found that we'll get to is that they have contrasting elevational distributions. And so this work we recently published in Forest Ecology and Management with Chloe Wassany's former master's student, Franklin Alonji, who used to be a former undergrad and master's student, and Sean Hoy-Skubik, a current PhD student. And so what we were kind of building on was observations made by Andy and others that whitebark pine kind of inhabits kind of upper elevations in the GYE, and limber pine can inhabit both upper and lower elevations. And what we wanted to know was, okay, why is this? Are there any traits at the juvenile stage that may link to these contrasting elevational distributions? So we want to understand these mechanisms, these physiological mechanisms contributing to whitebark at higher elevations, or what we call a specialist, and limber pine at kind of both high and low elevations, and why we call it a generalist. And we will talk more on this. And so generally, we were able to measure a suite of traits and kind of allocate them to this continuum of kind of fast traits or fast growth traits and slow growth traits. And hypothesized and found that kind of the specialist or whitebark pine really specialized in having these slower, more stress-resistant, survival-focused traits, whereas limber pine, the generalist, exhibited traits that were both stress-resistant, but also acquisitive or kind of promoted faster growth than whitebark pine. And so we found that this is kind of the takeaway overarching summary slide of, whitebark and limber are good at occupying these upper elevations. They're also very stress resistant. They're slow growing, and they had similar traits that showed this. Additionally, limber pine appears to be a little faster growing than whitebark pine because it has a greater ability to acquire sunlight, water, and CO2 compared to whitebark pine, which may be contributing to to our observation of limber pine as a generalist and possibly its ability to inhabit both lower and upper elevations. And then now we're going to focus on just highlight each grad student's work very, very briefly. So first, we'll start with PhD student Sean Hoy-Skubik, who started off in the lab as a master's student, now transitioned to a PhD student, but started off asking the question, how do these 3 species, whitebark, limber, and bristlecone, respond to heat and drought stress? And as a lot of you know, we call ourselves plant torturers. We're really good at torturing plants, killing plants, droughting them, heating them, and kind of measuring how they respond physiologically. And so these are some photos of Sean's from a drought experiment, where we could see that one was droughted and what Sean lovingly calls crispy, and then also a happy plant that was not drought stressed. And Sean measured a slew of physiological variables, but one I'm going to highlight today, and that kind of leads to your kind of current work now, is Sean developed and measured non-structural carbohydrates, which is a specialty of the lab that would not be possible without Sean's hard work getting that assay up and going. So Sean measured these carbohydrates, which are products of photosynthesis. They're used for various plant functions, stress resistance, and survival, and found that we measure starches and sugars, these types of carbohydrates, and found that pre-drought starch was kind of a predictor of whether a tree will survive or die during drought, which is pretty cool. So Sean found that crispy plants had lower starch pre-drought, and found that these happier plants that were able to survive a drought had higher pre-drought starch levels. And so if we're able to measure carbohydrate levels before a drought, then we may be able to predict whether an individual will survive or die of drought, which is pretty awesome. And recent work has shown that you can remotely sense these carbohydrates using remote sensing tools. And so that brings us to kind of Sean's kind of current focus right now and project focused by Montana NASA EPSCoR with collaborators Scott Powell and Anna Schweiger with the goal of remotely sensing pre-drought plant carbon dynamics to predict drought survival and mortality. So very cool project and cool direction. Katie Sparks is a master's student who is focusing on whitebark pine and also another 5-needle pine, foxtail pine. And she's obtained foxtail pines from 2 distinct populations in California, a northern population and a southern population, and wanted to look at how do these populations and species physiologically respond to drought. Katie developed this new drought method where we kind of use put pots on these floral foam blocks to use capillary action to create a slow, kind of moderate drought. She measured, so she used this new method that we hadn't used before to implement a drought, measured various physiological measurements, like using the Lycor to measure photosynthesis, stomatal conductance shown here. Katie's also measuring volatile organic compounds, or VOCs, in collaboration with Justin Runyon with the Forest Service, and comparing those among those populations and species. And Katie also developed this really neat protocol for looking at xylem traits, or that water conducting tissue of trees. And so this is a cross section of a stem of a whitebark pine. And from these images, we can use computer software to measure different traits of this water conducting tissue that may link to drought resistance of northern and southern foxtail populations and between foxtail and whitebark pine. So lots of really cool work. Next is Jess Harris, who's a master's student as well. And so we've been talking about these kind of juvenile seedlings or juveniles. They're kind of 2 years old, 5 years old. What Jess is, one of her projects is focusing on is kind of first year germinant seedling stage. So she obtained a bunch of seeds, small seeds, whitebark pine and limber pine, from different seed zones throughout the Pacific Northwest. So from throughout Oregon and Washington shown here, and also British Columbia, not shown here. And she planted these in the greenhouse from seed and measured various phenological and growth traits within this first year of growth. So something we had not yet done. You kind of ask, how do whitebark pine seedling traits, phenological developmental traits differ between seed zones? And so one of the traits that she measured was emergence. So kind of the percent of seeds that emerge, which I'm kind of showing here. And some preliminary data that we just talked about a couple of days ago that I was excited to share is this plot here of elevation, and each dot is a location from where we obtain seeds of whitebark pine, and then emergence rate and percent on the y-axis. And we find a pretty an interesting negative relationship between elevation and this emergence rate. So higher emergence from seeds sourced from lower elevations. So exciting preliminary data that we're still chomping on right now. Okay, so we're focusing on seedlings, juveniles, really small seeds. Now we're going to scale up to kind of mature trees. I want to highlight Tio Rautu's work, funded by the Montana Water Center, and kind of led by David Thoma, Brian Smithers, Aaron Shanahan, Mike Tursick, where Tio wanted to examine, with declining whitebark pine populations due to a suite of threats, how does that impact stream flow and snowpack? There are lots of studies, and when we write proposals, we say these high elevation pines protect snowpack and prolong stream flow, but it's based on early and rudimentary studies that haven't adequately tested this. So that's what TO set out to do with a great team and a great toolbox of tools. So TO's selected sites in Hood Creek in Highlight, in Beehive Basin, on this side, Big Sky, where she's measured various metrics of stream flow, also canopy cover using this neat canopy app. And it's also combining those kind of field-based measurements with a water balance model. So to answer that question, okay, if white bark pine is declining due to bark beetle, white pine blister rust, how is that affecting drivers of stream flow and snowpack? So here are some photos that Tio shared of, I believe this is a highlight among some pines, some dead pines. Making some snow measurements and also some stream flow measurements. And then finally, before we get to Lou, master student Steve Heisman is working on more of a mapping project. And this work is led by Brian Smithers, David Toma, Aaron, and Mike again. And the goal is to look at climate and water balance thresholds of white bark pine currently and map those thresholds, those kind of climate abiotic stressor thresholds, along with the prevalence of disturbance agents of white bark pine. So white pine blister rust, wildfire, drought, bark beetle, and kind of overlay all of those different maps to be able to come up with a useful management tool to map white bark pine's vulnerability to climate change and global change and identify good kind of planting sites and refugia in the Greater Yellowstone ecosystem for white bark pine. So kind of using or developing this tool to better identify suitable planting locations. So lots of cool work going on. And then with that, we'll wrap up and segue to Lou and let her share about her project specifically. So,
thanks, Danielle, for that introduction to everything that our lab studies and works on. And thanks everybody for being here. As Danielle mentioned, my name is Lou Duloisy. I'm a second-year PhD student in the Ulrich lab, studying the physiology of two high-elevation pines, Pinus albicaulis, commonly known as white bark pine, and Pinus flexilis, commonly known as limber pine. We're going to start off with a little activity. And looking at these two pictures that I took from my field sites, I want us all to test how well we can identify white bark pine and limber pine. So first, I want to see a show of hands as who has seen white bark pine and limber pine in the field. Great. So a fair bit of us have seen these trees on our adventures, probably in our own backyard. We are going to start with the picture to your right, and I want you all to take a few moments and think about whether you think that this is a white bark pine or a limber pine. So take a few moments, feel free to turn to your neighbors, and then I'm going to ask you all to raise your hands again. So potentially that felt a little bit hard. It also feels hard for me when I look at these pictures or I'm in the field. The one with the pink tag is a white bark pine, and the one without the pink tag is a limber pine. So we were somewhat correct. But the point being that identifying white bark pine and limber pine in the field when the seed cones aren't present, that kind of characteristic purple closed seed cone of the white bark pine isn't present, is incredibly difficult. And because of this, research on mature white bark and limber in the field has been challenging or hindered up until recently. Thanks to work by Dr. Lavin and Dr. Hansen that has been mentioned a bit up until this point, they created a relatively simple and economical method to determine if white bark pine and limber pine are genetically white bark pine or a limber pine, which I'll loop back to a little bit later in this talk, but I wanted to do a small activity before I hop in. So white bark pine and limber pine have overlapping distributions in the Western United States, with white bark pine having the largest and most northern distribution, and limber pine having a more southern distribution. But if you all take a look over to our neck of the woods in the Greater Yellowstone ecosystem, you'll notice that they have a large overlap distribution. So as Danielle and Andy have mentioned a little bit, white bark pine and limber pine are considered foundational and keystone species. And as such, they provide some invaluable ecosystem services in the ecosystems that they occupy. Clark's nutcrackers, for example, disperse and cache their seeds. Their seeds are a high nutrition food source for grizzly bears, and their ability to establish in kind of steep, windy areas provide habitat for animals such as grizzly bears and black bears and other small mammals. In addition, they provide a lot of snow stabilization and soil stabilization, which is a critical watershed protection. However, both of these species are currently facing a suite of threats, including mountain pine blister rust and the mountain pine beetle and severe drought. These threats are expected to continue as we see warmer and drier conditions, which will continue to exacerbate the widespread mortality that we're seeing. Because of this, whitebark pine was listed as threatened under the Endangered Species Act as of December 2022, so relatively recently. Because of the niche that these species occupy in their ecosystems and also the multifaceted threats that they're facing, it's critical that we understand the physiological mechanisms underlying how and why these species currently inhabit their environments in order to better predict how climate change will impact these species and to protect these species. So making this link between how important it is to understand the physiology behind species distributions. So whitebark pine, as previously mentioned by Danielle, occupies a narrower distribution elevationally between 1,600 and 3,600 meters, while limber pine occupies a broader elevational gradient, about 870 to 3,800 meters. In the Greater Yellowstone ecosystem specifically, whitebark pine has been documented at higher elevations, while limber pine has been documented at lower elevations and in this broader elevational gradient. You can see on this graph by Sean Hoyes-Cubic from FIA data that the whitebark pine elevational gradient is much more squished, indicating that higher elevation, while the limber pine is more broad. And because of these elevational differences in elevational distribution, Dr. Tomback and other previous researchers have defined whitebark pine as more of a specialist and limber pine as more of a generalist. Additionally, previous work in the Ulrich lab has quantified some physiological traits that are concurrent with the functions of a specialist whitebark and a generalist limber pine, but in seedlings in the greenhouse. To date, there's still a large gap in the research in field studies of mature whitebark pine and limber pine. And these field studies are critical for understanding how our changing climate relates to species distribution. Directly into the research questions that we decided to ask because of this gap in our research. We asked, how do seasonal patterns of physiology, so leaf water potential, gas exchange, and non-structural carbohydrates, differ between whitebark pine and limber pine? And based on previous research done by the Ulrich lab and other researchers I've mentioned, we hypothesized that seasonal patterns of physiology would indicate that limber pine would be less drought stressed than whitebark pine, concurrent with that specialist generalist differentiation. So we based this off of previous research by Dr. Lavin and Dr. Hansen that I mentioned, who developed a genetic technique for distinguishing non-seed bearing whitebark pine and limber pine in the field. They created 13 transects ranging from the lower tree line to the subalpine in the Greater Yellowstone ecosystem, where they identified genetically whitebark pine and limber pine. And out of these 13 sites, we chose 2 sites based on our criteria to assess our research questions. So our 2 sites were Mile Creek and Teton Pass. We chose these 2 sites because they had overlapping whitebark pine and limber pine, and additionally due to just access from Montana State University, because we wanted to visit these sites several times. And the last determining factor was acquiring permits. With the recent listing of whitebark pine as threatened under the Endangered Species Act, it, as you can imagine, increased difficulty getting permits to collect samples in whitebark pine. When I reached out to land managers in March, just a couple months after the listing, there were not yet protocols in place for issuing permits for whitebark pine. So we narrowed it down to 2 sites based off of those 3 reasons: Mile Creek and Teton Pass, both with co-occurring whitebark pine and limber pine. You can see the whitebark pine is shown in the red circles, and the limber pine is shown in the blue circles. Mile Creek had 25 total individuals that were genetically identified, and we chose 10 individuals, 5 white bark and 5 limber, to make all of these measurements on. Teton Pass had a similar number, 23 total individuals, and we chose 14 individuals for the physiological measurements, 7 white bark and 7 limber. We identified these individuals using transects by Hansen et al. And we wanted to control for elevation, so we kept all of the individuals that we studied within 1,000 vertical feet of each other in order to assure that the differences we were seeing were truly species differences and not differences in elevation. So each of these sites were visited 4 times throughout the summer on what we coined as the growing season. So June, July, August, and September. The site visits took 3 to 5 days and required a bit of a backcountry sampling plan because these sites, especially Mile Creek, required a 3-mile hike in. And then Teton Pass, although there was close proximity to the road, also required quite a bit of hiking and elevation gain. Briefly, we also wanted to note that there was no significant differences in climate data between these 2 sites. This is the 2023 PRISM data for Mile Creek in green and Teton Pass in purple. There's no September yet because September is not over, but there aren't any drastic differences between precip, temperature, and vapor pressure deficit. So at each of these 3 sites, we looked at photosynthetic performance using the Lycor, which Danielle mentioned earlier. We generated ACI curves for each individual. We have an example here on the right from an individual at Mile Creek. And I just wanted to briefly talk about some parameters that we can use from these graphs that tell us about photosynthetic performance. One of the parameters, the initial slope here, is VC max, which is the rate of carboxylation, which tells you about the photosynthetic capacity per unit leaf area. The second component I wanted to mention is A max, which is the maximum amount of carbon a plant is able to uptake during photosynthesis. So each of these parameters, VC max and A max, tell us about the photosynthetic performance of the individuals. We have some very preliminary results here because I just finished my field season on Friday. So here we have VC max, which is one of the photosynthetic parameters that tell us about performance. We did not observe any significant differences during the month of June between white bark pine and limber pine at either site. However, based on previous research and concurrent with our hypotheses, I am predicting that once I analyze the data for July, August, and September, white bark pine will have a higher VC max than limber pine, and we will start seeing those species differences with drier and warmer temperatures. So this is very preliminary. Another metric we looked at was leaf water potential, which is a metric that helps us understand how drought stressed a tree is. So leaf water potential is taken at 2 time points, pre-dawn and midday. The pre-dawn time point helps us understand how much water a tree has available when it is at equilibrium with the soil, while midday water potential helps us understand how much water is available when evapotranspiration is happening or other stressors that are stressing that water column. And it's important to take both so that we can look at either and also look at the changes throughout the day for any given tree. Sampling for leaf water potential pre-dawn that needs to happen an hour before sunrise occurs. So what that ends up looking like is us getting up at 2:30 in the morning, as you can imagine in those early summer months when sunrise is at 5:30, and hiking out and sampling. So thankfully I had some amenable field techs who maybe weren't super excited, but were happy to go out and do that with me. Again, some preliminary results for pre-dawn water potential, which is the metric we took an hour before sunrise. Just to orient you to the axes, a more negative water potential indicates a more water-stressed tree. You can tell by what Sean would call a crispy pine tree at the bottom. And a less negative water potential indicates a less water-stressed tree, again depicted by a happier pine tree. Similar than with the VC Max results, we did not see any significant differences in June, July, and August between white bark pine and limber pine. However, again, concurrent with our hypotheses, I'm excited to look at the September values. They looked a lot more negative upon first glance. And I'm predicting that because of white bark pine being more of a specialist, its water potential values will become more negative as it becomes more stressed in drier and warmer conditions later in the summer. The last measurement that we took is we collected samples for non-structural carbohydrates. Non-structural carbohydrates are the primary product of photosynthesis, and plants rely on both newly assimilated carbon and stored non-structural carbohydrates for growth in a variety of physiological functions like respiration, osmotic regulation, and defense. And something that the Ulrich lab specializes in, especially thanks to Sean and Danielle, is looking at non-structural carbohydrates and how plants allocate their carbon during stress. So I was excited to look at this on mature trees out in the field. Because non-structural carbohydrate analysis requires lab work, and I just finished my field work on Friday, we have not completed the lab work yet. However, I'm predicting that because of limber pine's more generalist strategies, it's going to have a higher percentage of non-structural carbohydrates as it is less drought stressed than white bark pine. Trees often use non-structural carbohydrates during stressful conditions. And so if white bark pine is more drought stressed, I'm assuming it's going to have less, or I'm predicting that it's going to have less non-structural carbohydrates. Sean, in his previous work in seedlings in the greenhouse, has found that limber pine does have higher non-structural carbohydrate content in seedlings. So I'm looking forward to seeing how this looks for our trees in the field. So moving forward, I want to revisit our hypotheses that we expected that seasonal patterns of physiology will indicate that limber pine will be less drought stressed than white bark pine. Although we have yet to see this in our early summer months, I am predicting that as I get this data for the later summer, white bark pine will have patterns of physiology that indicate it being more stressed than limber pine. And bringing it back to this point, I haven't analyzed this data yet, but on first glance in September, the pre-dawn values were so much lower, we ran out of our nitrogen in half of the time. So I'm looking forward to seeing how those values compare to the other months. And I'm hoping that those, or I'm thinking that they may reflect drier conditions and also some species differences. The next steps for this project, as I keep hinting at, are continuing to analyze this August and September data, and then revisiting the sites next summer. We had a particularly wet summer, and I think it will be exciting to look at yearly differences in physiology. And then I wanted to wrap it all up with this, thinking that it is absolutely critical that we understand the physiological mechanisms underlying how and why these two species inhabitate their environments if we want to understand their distributions in our changing climate. And then lastly, this work would not be possible without the help of three incredible field techs, Zoe, Sarah, and Oscar, who thankfully were willing to get up very early with me and hike up Teton Pass, which if you've ever been there, is quite a feat at 3 in the morning, and also carry some relatively heavy equipment. If you've ever used a Lycor, you know that it's not actually very light. And so they are the village that makes this research function, and also our funding sources, the Montana Institute on Ecosystems and the White Bark Pine, and then lastly, the Ulrich Lab, Danielle, and all the grad students in it who have been around to help me bounce ideas off of them and hear me talk about my field adventure.
I guess I have a question. Do you guys take any measures of like to measure drought, I guess, at the specific field sites, like anything in the soil, or is it just based off of atmospheric and kind of remote sensing data?
Yeah, that's a good question. We didn't take any like spot measurements at the site. However, I'm looking, the climate data that I showed you was from Prism, but both of these sites actually have a climate, like Teton Pass has a SNOTEL site that also puts out daily climate values. And I'm excited to kind of dive into how that looked more specifically than the Prism data, but no actual spot measurements for soil moisture.
Really cool stuff overall. I Riffing on that comment, the work is really, you guys have discussed so far, focused in on responses of the two species. You conducted any experiments to determine like thermal optima? You know, not just drought, but temperature, other environmental variables, like where do they do best at certain ecophysiological, in a common laboratory setting, perhaps? And then related to that, the curves you show of the distribution and with elevation, you know, where there's overlap, you imagine that elevation as a resource, which of course a lot of resources change along elevation potentially. It really, you know, it really strikes you with a classic sort of coexistence, you know, is that resource gradient, and have you guys performed or thought about experiments where you have them duke it out to see who survives? To like, why do both of those species exist? Why isn't one?
Yeah, that's a great question. I think those questions are really maybe better suited to a greenhouse experiment, which Sean and Danielle have done where you have both species and you're putting drought or like inciting drought on them. I don't know if you all want to ping in.
Yeah, for your first question, Jack, on the thermal optimum, we do temperature response curves on foliage. So we've done a lot of cold tolerance and heat tolerance on foliage. We would kind of expose excised needles to various temperatures in a water bath and are able to create those curves. We have not connected kind of that leaf level thermal optimum to, or mapping it out, I guess, to know specifically, okay, at the leaf level, this is the thermal optimum. How does that actually relate to the whole plant thermal optimum? And so that's, that kind of links to one of Sean's experiments where he did do a heat stress, where he heat stressed whole plants, whole seedlings. And so maybe connecting like the leaf level, heat and cold tolerance curves, whole plant temperature responses, maybe we can get at that, but we haven't mapped that out. I think that's what maybe you were getting at there. And then, yeah, there's so many good questions in what you mentioned. My other thought, what's the second part?
There was just a curiosity about this sort of whether you could apply a sort of coexistence framework to this, and it may be an inappropriate system to think about it like that, but.
My first thought, and maybe Brian could join in too, if you're interested, but is it maybe what might be limiting could be a resource, but it also, especially in this mountain ecosystem that I know Brian spends a lot of time in, is temperature. And so maybe at a whole plant level, white bark pine is very conducive maybe to its colder temperatures, or maybe it's less conducive to warmer temperatures. I'll put it that way, because we've got limber pine at both of those very, very high elevations too. And then the other part that Andy's done some work into is with competition between white bark and limber, maybe at lower elevations, which could link to their differences in ability of white bark to be tolerant of drought and heat than those lower, more arid elevations. I don't know.
Yeah, that makes sense.
Just relatedly, it's thought that the main competitors are not each other, but rather each species with other conifer species, Douglas fir in particular, lodgepole, subalpine fir. And so now that leads to, we need all the same studies done on those other species that might be competitors and might in fact explain these distributions to some extent.
BC max, is that what, is that the right thing? The figure you had, which had the really big distribution, or a lot of variation in, yeah, I think we're real close. That's the one. And I really totally understand. You said this is preliminary, sort of just getting in there. I was sort of taken with obviously the difference in variation in white bark pine versus limber pine. Is that, if that holds, and because I do not understand physiology well, what would that mean if there's greater variation in VC max relative to limber pine?
Yeah, That's a great question. I was also taken aback by that, mostly because I thought that white bark pine, being more of a specialist, would have a much narrower range of values. This holds. I think that it would show that within white bark pine individuals, this isn't really saying much, but a much higher range of the rate of carboxylation and how much photosynthesis can occur on a unit per area basis. But yeah, I'm excited to see for July, June and July, like all of them together, and see if this is maybe just, this was our first month out there using the Lycor in the backcountry. And it looks a little funky, or if this is how it's gonna look for each month.
It's measuring the slope of that curve.
Yeah, so the VC max is this original slope right here.
Okay, so it's like how much more assimilation can happen with an increase in.
Yeah, so this original slope is showing how much carbon assimilation can happen until the plant is unable to assimilate more carbon based on more cellular processes. So what's happening here is as we input, the Lycor inputs higher rates of carbon. And in theory, at first, the more carbon you input, the more photosynthesis can occur until you reach that curve where cellular processes prevent just like infinite amounts of photosynthesis to occur. So the VC max is that initial slope.
I would add to your point, Brian, maybe, and this is something Lou and I can talk about for next summer, next field season, if there is that variation in VC max, for example, maybe other things in white bark pine, maybe sampling, white bark at lower elevation and higher elevation, maybe would we see that greater elevation in that way? If we're seeing that within kind of this relatively similar elevations, maybe to really get at that, see the differences there. I mean, maybe we don't see that in limber pine, but yeah.
Mm-hmm. Cool.